what someone said to you? It's in your genes. They weren't talking about your pants. Ha! No, they meant you were born that way. It's in your genes. Now, genes tell your cells what to do. Like, you're a stomach cell. You're a brain cell. You're gonna have brown eyes and a big nose. Now, genes are made of DNA, deoxyribonucleic acid.
Since DNA is the basis of life, it makes sense to study the thing. However, sometimes there ain't enough DNA to go around. Sometimes we just have one piece of rare DNA that we can't afford to mess up. That's why we have PCR. It is a method that helps us make many copies of just one DNA molecule. PCR stands for polymerase chain reaction, since it uses the enzyme DNA polymerase to essentially cause a chain reaction that will make the copies of just that one DNA molecule.
Everything's a copy of a copy of a copy.
The process of PCR is simple. We break apart the DNA molecule into two strands. Then we stick some special bracketing molecules that will essentially mark the starting position and the end position of where the enzyme should go. Then we let the DNA polymerase do its job. In a short time, we can have billions of DNA clones from just one molecule.
PCR begins with denaturization. 5 components are added to a mixture of base primers, the original DNA, which will be called the template DNA, tag DNA polymerase, and the necessary buffers for the reaction to go without problems. Once the solution is prepared, PCR can begin. The temperature is raised to 95 degrees Celsius. At this temperature, the template DNA strands come apart since the hydrogen bonds between the nucleotide bases are broken, forming 2 template strands with their bases exposed. As long as the temperature is high, the components of the mixture won't react, helping keep As long as the temperature is high, the other components of the mixture won't react, helping keep the next step separate.
The 2 template strands are ready to be copied. However, we are only interested in a small portion of each strand, not the whole thing. To restrict where the copying process should begin and where it should end, we have to use primers. Remember, primers are tiny pieces of special DNA that are designed to stick to certain spots on the strand where each other's bases match. The primer has a certain sequence. The sequence will match with some spot in the DNA strand. Once it finds the match, it will stick to the DNA strand and show the enzyme Taq DNA polymerase where to start. Primers work in a lower temperature, so the mixture to be cooled down to 55 degrees Celsius. In these conditions, the primers will stick to the DNA strands at the spots that match it. The step ensures that the primers flank the position of the DNA that needs to be copied.
I see a red light.
And go.
Does that mean it's on?
Yes.
Are we on air?
Yes, go.
Are we on air?
And go.
Once the primers are in place, the temperature is raised again to 75 degrees Celsius in order to activate the enzyme Taq polymerase. This enzyme can be used to create new double-stranded DNA molecules from the 2 original strands. It does this by first attaching itself to the spots where the primers have flanked the special DNA. Then it moves down the DNA strand and attaches nucleotides that match with the strand sequence. The polymerase gets nucleotides from the surrounding mixture, grabs the ones it needs, one at a time, and attaches them all along the line. The Taq polymerase attaches the nucleotides in such a way that it creates a brand new strand that is attached to the old strand, therefore it's DNA synthesis. And Shabazzel and Shaban, the 2 copies of the original DNA sequence. In order to get the specific sequence that we want, PCR has to be repeated many more times. This is to ensure that we amplify the exact piece of DNA that we want to study. With each repetition of PCR, the DNA sequence that contains the gene that we're looking for is made with more abundance until we get millions of copies of the sequence. we want to study. With patience and some proper tending to the samples, PCR can produce many clones of the DNA of interest, to where you can have a sample with 99.9% concentration of the gene of interest. Where you once had one DNA, you now have billions. Suck.
Chicken nugget, biscuit nugget in a biscuit nugget, biscuit nugget in a biscuit nugget, biscuit nugget in a biscuit. Dip it all in mashed potato nugget, biscuit nugget in a biscuit nugget, biscuit nugget in a biscuit nugget, biscuit nugget in a biscuit. Dip it all in mashed potatoes. Dip it all in mashed potatoes. Dip the mashed potato gobble chicken nugget biscuit in the barbecue sauce. Yum, yum, gimme! Biscuit nugget in a biscuit nugget, biscuit nugget in a biscuit nugget, biscuit nugget in a biscuit. Dip it all in mashed potatoes. Biscuit nugget in a biscuit nugget, biscuit nugget in a biscuit nugget, biscuit nugget in a biscuit. Dip it all in mashed potatoes. Dip it all in mashed potatoes. Dip the mashed potato gobble chicken nugget biscuit in the barbecue sauce.